Okay, awesome. Thank you. So welcome. Good evening, everybody. Good morning, good afternoon, wherever you are. It's evening my time here on Thanksgiving Day in San Francisco. So I am going to be your host for the first part of the session here today. My name is Baljit Yubi. Really wanted to introduce you to the virtual Podium 2020 Asia Pacific. And if you've joined any of the prior sessions, you'll know that this platform was actually established to promote scientific engagement and discussion, collaboration, and networking in the field of metabolomics and lipidomics during the time of the pandemic, where we've lost the face-to-face ability to do this. So Virtual Podium 1.0 happened earlier this year in March, and it ran all the way through to May up until ASMS 2020. And then this was put together by this amazing team of people here that you can see on the slide. And it was actually put together for the Asia Pacific region. And again, the goal is to create that sense of community, continue the networking, continue the sharing of science. And we have been meeting every Friday to hear lightning talks, 5-minute lightning talks, as well as some really amazing keynote talks from thought leaders and people within the field of metabolomics in your part of the world. So next slide, please. So this is the agenda for today. So we've got a really great agenda. Today is the last of the 4 sessions. So if you've missed them before, they are recorded and they're up on YouTube, and we can share the links with you. We have 3 great keynote speakers today, and Professor Peter Meikle will be closing the session today. And so you can see a list of all the lightning talks that are listed here. And as I said, 3 amazing keynote speakers for you today. Next slide, please. Yeah, these are our lightning talks from the individuals and the institutions. So we have 5 of them today. And this is where they're from within your region. Next slide, please. So it's my honor, and I'm really proud to introduce our first keynote speaker for the last session today. And his name is Professor David Broadhurst. And he is a name that you should all know by now in this field. I mean, he actually started his professional career under the guidance of Professor Douglas Kell and Roy Goodacre. He's been a very active member of the community for over 20 years. He's pioneered various works and has published in the literature very, very readily. He did his postdoctoral research studies at the University of Manchester, and actually developed gold standards alongside Warwick Dunn around clinical metabolomics protocols. He moved from there to Cork University, and then in 2011, he was appointed the Associate Professor of Biostatistics at the University of Alberta in Canada. In 2016, he was appointed the Professor of Data Science and Biostatistics at Edith Cowan University in Perth, Australia. That's where he is today, and he's helped establish the Australian National Phenome Centre in Western Australia before settling in and setting up his own in-house facility. So it's with my pleasure to turn over the floor to Professor David Broadhurst, who's actually going to be talking about the inherent dangers of relying on pooled QCs for post-hoc assessment of measurement precision in untargeted clinical metabolomics. The floor is yours.
Okay. Can everybody hear me okay?
Yep, we can hear you.
How do I share? There we go. Okay, can everybody see the slides?
Yep.
Okay, so this is actually, hold on a sec. This is actually a short talk in response to my own paper, which was published back in 2018. This was a fantastic collaboration between my lab in Perth, the Imperial College Phenome Centre, the Birmingham Phenome Centre, and then Roy Goodacre Manchester University is now at Liverpool University. And we talked about the use of pooled QC samples and kind of describe the current gold standard, if you like, for doing that. It's been a reasonably popular paper, and it's good that people are enjoying reading it. So, just as there's going to be people in the audience with an analytical chemistry background, and this slide should be very familiar for those people, but equally, I imagine there'll be some epidemiologists in the audience, and they maybe have not really got to grips with the way technology works. So if we just initially look at a targeted quantified metabolite workflow, this is a very simplified overview, but if you imagine the idea is we're just looking at one metabolite here. We have a certain concentration of a metabolite in a given matrix, say blood or urine. Then that is applied to a measurement system, which could be a mass spectrometer or an NMR system. And within that measurement system, I'm including the concept of doing all the sample prep that you would do before you apply that sample, inject that sample into the instrument. The instrument will then produce a response, which in mass spec will be a peak area, certainly in LCMS. And then in order to convert that peak area into a numerical concentration, there's a calibration system, which is normally a calibration curve, which is a mathematical function which converts peak area into the concentration of the analyte. Now, let's just talk a bit more about calibration curves. So a calibration curve is just a graphical representation of an equation. So we've got a very simplified calibration curve. It's a linear regression, and we're mapping the metabolite concentration to the peak area. Now, how you build that calibration curve in a targeted assay is you would inject known concentrations of a metabolite, record the peak area, plot those points, draw a line, and then calculate the slope and the intercept of that line. And that will give you the equation which will map concentrations to peak area. And then the inverse of that function will map peak area back to concentration when you've got an unknown peak area. Now, a calibration curve can be more or less sensitive. It depends on the metabolite, it would depend on the instrument. A less sensitive assay for a given range of metabolite concentration, you'll get a small range in peak area, peak response. Whereas a more sensitive instrument, for the same change in metabolite concentration, you'll get a much larger peak area. And so you can clearly see from these two arrows here that the variance in response 1 is a lot less than the response 2. Now, an alternative to the change in sensitivity or the change in gradient is actually a shift in the limit of detection. And this is pretty much the idea here is that regression line is shifting along the axis there, and that will change where the mean peak area sits. So again, if we just do two simple examples, same concentration of metabolite, and then for response 1, we've got that range, and then response 2, we've got this range. Now, the variance is the same. All that's different is you've got a shift in the mean value. When you convert back, if you use the right calibration curve, then you'll get back to the correct concentration. So that's a very simple overview and a perfect system. Of course, every measurement instrument in the world, even NMR for the people who get excited about the reproducibility of NMR, every instrument has noise. So it's not as simple as just what I've described. And the best way to visualize that noise in the system is to create a fuzzy calibration curve. And here, when we apply a particular metabolite concentration, there is a random chance that it will be somewhere in between these two boundaries and clearly more likely to be in the middle. So this is if it's a Gaussian noise, then this is a Gaussian distribution, and therefore it's a random variable. So there's a probability of your measurement being sitting between those two areas. So in order to determine how precise your instrument is, the standard method is to inject multiple samples of the same metabolite concentration into the system, and then you will calculate the standard deviation of the peak areas that come out, and then you would normalize to the mean so that you can standardize across different assays. And that is where we get the percent relative standard deviation. And percent RSD is a standard measure for precision. It's used in targeted assays, and it's used extensively in untargeted metabolomics. Now, you can reverse this process as well, and for a given peak area, you can use that fuzzy calibration curve, and you will get a metabolite concentration, but which will also have an error component. And the important thing to note here, and this is very important, and I'll come back to this right at the end of my talk, the RSD for the peak is not the RSD for the concentration. So when you have an assay which is described in terms of concentration and a precision in concentration, it is not talking about the precision in peak area. Now, of course, in metabolomics, we don't have a calibration curve. And we replace the calibration curve by deconvolution algorithms that group and align the peaks so that we can then use that data. But the output that we get is actually still peak areas, and there is no calibration, so we don't actually know the actual quantified amount of that metabolite. Everything is a relative quantification. Now, we can actually consider that whole process as a single transfer function, where we're converting the concentration in metabolite X, which we won't necessarily know, into a peak area, and the error component is a combined error component for the whole system. Now, how do we go about determining precision for untargeted metabolomics when we don't use a standard assay and we don't have inject standard amounts of metabolite concentrations? What we end up doing is using a process which was first developed by Ian Wilson and his team back in 2006. And this is the idea of a pooled QC, where you take a small aliquot of each of the samples in your study, mix them together, and then sub-aliquot them out, and then periodically inject those QCs, the red squares here, through your batch, so that you can track the precision over time, and then you can calculate an RSD for that batch by using those data points. Now, when Rick Dunn and I joined Ian Wilson and we started working on the HughesMet project, which was led at Manchester by Douglas Kell, we started investigating this further, and we soon realized that without that calibration curve, the instability in the instrument was actually quite extreme. And then across batches and multiple batches, we were actually getting significant variation in that peak response. You can see here on the left, this is the need for conditioning your column if you're using an LCMS system or a GCMS system. And here you can see how when we injected a QC across 120 samples, how that, in a PCA space, how that response changed over time. And there's clearly a very systematic shift in the peak response. And then if we think about it in terms of number of batches, you can see that there's a batch drift. Now, if we look at within a single batch, we actually started seeing very systematic bias within that batch per peak. And each peak might have a completely different trajectory. In this case, we've picked a particularly terrible nonlinear drift, but it's not we're not able to use a generalized across multiple peaks. Equally, we just saw that there's a significant batch effect between multiple batches. So here we've got 3 batches in which the second batch has a really bad drop in response. Now, it turns out during the HughesMet project that there's actually a way to get around this, and I developed a piece of software using cubic splines to actually model that drift whilst not removing the noise in the data so that we could still have a fairly accurate measurement. And then as a 2-step process, first you correct the within batch variation, which might be nonlinear. Once that's corrected, then we can correct the between batch variance in a very simple way as illustrated in this plot. So that ultimately ended up with our Nature Protocols paper and a paper which talked about the need for experimental design where we have careful randomization within and across batches so that we don't introduce any statistical bias in our biological interpretation. And if we do some reasonable data cleaning and use these methods, the correction methods, you could start seeing that we can get very reproducible metabolomics data, which is shown in this PCA spot rotating over on the right here. As a more recent example, this is from 2017, and I worked closely with Craig Wheelock at the Karolinska Institute, and he had a study where he was looking at data over 17 batches. So we successfully used my algorithm to correct the within and between batch variation. You can see that that's his data was fantastically clean. The QC was wonderful. So you can see that the algorithm works spectacularly well on this data. And you can see that we go from something which would be unusable statistically to something that's really quite good. So this got me thinking. So that's the state of play at the moment. That's what we describe in the paper at the start of my talk. But I've started to think over the last 3 or 4 years, why is this happening? Why is that drift happening? What is the underlying maths? What is the abstract concepts that we need to think about? And from this part of the talk on, we really need to take a bit of a leap of faith, because remember that metabolomics, untargeted metabolomics, we don't have an actual calibration curve. Well, there's a calibration curve there, but we don't determine it because we don't know, we don't have a set of standards for each of the peaks, because we don't know what the peaks are until the experiment's finished. So although there's always a calibration curve there, it's implied. And as such, we don't have this metabolite concentration. All we have is the peak area. So what I'm going to propose now is you to take this leap of faith with me to start thinking about the hypothetical calibration curve that's actually in the data or in the response of the instrument for a given study. And let's take the simple example first. We've got 2 batches here on the right where there's clearly a step change in the response, and then after using the correction algorithm, we can align those and they're usable. Note here that these are the identical samples that have been run twice in 2 batches, so they should align really well. But if you think of this in terms of a change in calibration curve, this is actually one of those first examples earlier on in the talk where there's a shift in the calibration where the gradient of the calibration curve doesn't change. There's just a shift from right to left. And that means that for the same range of calibration as metabolite concentrations, which of course we don't exactly know, we're getting 2 responses which are the same in terms of the variance, but are different in terms of the mean. So this is very, very easy to correct. We just write an algorithm which shifts that calibration curve or aligns those 2 calibration curves. And that's pretty much what the correction algorithm is doing. Now, that's known as additive bias. And if anybody was on Twitter last night, I was talking about my talk and I presented some animations. And my friend Nick decided that the reason why this happens is because of temporal crud. So basically what we're talking about is columns getting dirty, instruments getting dirty, and that chemical process is affecting the calibration curve, which is making that shift. Now, I just wanted to give a shout out to Nick and his new rock band. Now, this is far more insidious. What happens if we actually, rather than it being a shift in calibration in the axis, it's actually a shift in the gradient? And this illustrates here where we've got batch 1, which is quite a sensitive calibration, and batch 2, which is a less sensitive. And remember, these are hypothetical, so I'm just projecting this idea into this data space. And if you create data from a repeat of the same metabolite response into those 2 calibrations, you get this outcome. And these, again, are exactly the same data. We just got a different calibration curve. And again, we can correct that shift quite successfully. But the important thing here is look at the difference in variance. So this is actually unusable in terms of a statistical subsequent statistical analysis. So we have to be very, very careful about how we deal with this. And this is multiplicative bias, and this is something that nobody is taking any consideration for at the moment in metabolomic studies. So here's an within batch change, and this is one of the animations that I put up on Twitter last night. And what I'm showing here is that shift in calibration, but this happening over time within a batch. And you'll see that we're creating those metabolites here. They're being projected through this calibration curve to what the peak response is. And this is that peak response over time. And you can see over time, we're getting this linear shift. Again, you saw earlier in my talk, that is something that we can correct for quite easily, and the variance is constant. Now, what happens if we change the gradient? So we're projecting again up through this calibration curve, but the gradient is changing and it's expanding, and we're getting this amplification of the sensitivity and this coning of the data. And again, if you wanted to compare the data at the start of the batch to the end of the batch statistically, you're in danger of making some false assumptions here. So we can correct this, but we can't correct for the change in variance using a single pooled QC. So the important thing to think in terms of the system, that there's multiple points of noise within this system. And so therefore, these noise can be both multiplicative or additive or a mixture of both, but the system as a whole is a combination of the two. So we really need to be taking this multiplicative noise and bias into consideration. And here's a real world example again from Craig Wheelock's, and you can see here we did a really good job of correcting the data, but look, there's some coning going on there. Admittedly, it's slow and it takes a lot of injections to get that, but it's enough to disrupt the statistical interpretation. Now, the only way we could really get around that is by doing some sort of rotation and then scaling of the data. But we have to start asking the question, is this practical, and what sort of protocols could we enforce to do this? And will the mathematics get so complicated that it causes more harm than good? So in conclusion, first up, and this is really a subject for a second talk, is the RSD for a peak is not the RSD for the concentration. And we often just quote the RSD for the peak when we're talking about metabolomics and imply that it's actually something to do with the relative concentration, and it's not. So that's disingenuous. And I would actually suggest you go read my paper and start thinking about using the D ratio instead, which is constant across the peak and the concentration. Bias correction works well if there's no change in gradient. So if you inspect your data and there doesn't seem to be that change, fine, we can carry on as usual. The implications of this is that for small and medium-sized studies, and this is the size of study that I will only do now in my lab, pool QC seems to be fine because that nonlinearity or that change in gradient seems to be very slow and only comes into a factor when you use large-scale studies, so that's kind of dangerous. When we start looking at studies, multiple studies from the same lab, and then trying to link them together using standard reference material, again, if there's a step change in the sensitivity in that gradient, then you're not going to be able to join them together using just a single concentration reference material. The same goes for multiple labs. The same particularly goes if you're going to compare instrument manufacturers or different assays. It's just not going to work. So we have to be very, very, very careful moving forward when we're looking at untargeted metabolomics. The better approach is to take the approach of the epidemiologist and look at doing meta-analysis. Some people have suggested we could auto-scale per batch, but that is very, very dangerous as well, because you have to really guarantee that the variance across each batch is identical, or else you're going to make some assumptions about the data which will affect the subsequent statistics. So unsupervised methods for correcting for variation between batches, very dangerous.
About 30 seconds, David. Oh, there you go.
That's my last slide.
Yeah,
So
excellent. Perfect timing.
Craig Wheelock, Stacey and Harry from my lab, and Ian and Rick for their long-term collaboration. And everybody's welcome to come and visit Perth once the lockdown is over. And thank you very much.
Thank you, David. And the questions is open now for Professor David. There's one from Corey Gills from Baker IDI. He's asking, hi, David, I think you will notice that some groups are considering multiplicative versus additive noise. This is done by log transforming data before median centering. This turns multiplicative noise into additives. So.
No, I mean, I've looked at doing this as well, and my algorithm can do that, but that doesn't take into consideration the bias so much, because the interaction is complex. If you take the logarithm, so if you've got additive noise and then you take the logarithm of that, then that becomes, in the log form, a division. So the multiplicative noise turns into additive, and then the additive noise turns into multiplicative. So it's not as straightforward as just doing a log transformation.
Yeah. So there's another, thank you.
You'll notice on Craig's, you'll notice on the data from Craig Wheelock that the data is log transformed there, and you can see that coding is happening already, even when it's log transformed.
Yeah. So Clory is again asking, depends on how much you think additive noise is there. If you can quickly answer for that, there's more questions for you lining up.
Yeah, of course, but that's why we inject pooled QCs, and then that's why you look at the data. You don't have to correct for bias if there is none.
Yeah. So we know from by 21MA asking, thanks for a great talk. How many batches would you say are in a medium versus a large scale multi-batch study? Great question.
Well, I like to limit the studies we do in our lab to about maximum of around 300 injections. It starts to be diminishing returns in terms of the statistical power you're adding by having more samples to the noise you're adding by correcting for more batches and dealing with these sort of things. If you have a very solid assay, certain metabolites clearly that have a very invariant, not a problem. But if you start getting to RSDs that are around the borderline of 20%, then the more batches you do, and if you're going to try and align them, then you start adding in more noise than you're taking out.
Yeah. There's one more question. Yeah.
Tussie, as the super mean timekeeper, we'll probably have to move on, but we will have time at the end for specific questions, so people are encouraged to put them into the chat.
Okay.
Sorry.
No. that's okay. I'll share the screen now. Coming to the lightning talks. The first lightning talk by Igel Mellon. Igel is from Western Australia, University of Western Australia, and she's presenting lipid profile and cellular energy production in Australia. Igel, take away, and I'm going to share your slides.
Do I just ask you if I want to move on to the next slide?
Yes, please. Yeah.
Okay.
Let me get into. Sorry. Yeah. Can you see?
Yep. Okay.
All yours.
Hi, my name is Igel Malone. I'm a PhD candidate at the University of Western Australia, and my research is into the cellular energy production and lipid profile of mitochondria in 2 wheat varieties with differing heat tolerance. Wheat accounts for 1/5 of the global calorie and protein intake, and the demand for wheat and wheat products is increasing with the growing global population. High temperatures negatively impact on wheat yield, so the increase in heat waves that's the result of the changing global climate is a real danger to future food security, and identifying the molecular traits that contribute to heat tolerance in wheat is necessary. Cellular energy is essential for wheat growth and repair, and its production takes place in the mitochondria. This organelle features a phospholipid bilayer membrane embedded with respiratory complex proteins that carry out the final step of aerobic respiration. These membranes are sensitive to thermal stress owing to their lipid nature. However, the exact changes that occur in this lipid protein network are not well understood. Can I have the next slide, please? In my study, I looked at 2 wheat varieties that are reported to differ in their heat sensitivity at the grain production stage. So seedlings were grown at optimal growth temperatures, and the heat treatment was 2 days at 36 degrees daytime and 31-degree nighttime temperature. The fresh and dry weights and the height of 14-year-old seedlings was measured, and that's in the top right-hand corner of the screen. Maize is noted to be heat tolerant and EGA 2248 is noted to be heat sensitive, the significantly larger percentage reduction in biomass measurements in the heat sensitive variety confirms this, and it also indicates that heat sensitivity can be determined at a seedling stage. In the bottom left-hand corner, I'm moving from left to right, mitochondrial lipid profiling was conducted by targeted mass spectrometry using a triple quad mass spectrometer by either precursor or neutral loss scanning. 135 lipid species across 9 lipid classes were detected. Significant differences were found in the expression of 64 lipid species plotted on the heat map. The quantity of PC and PI increases during heat treatment, as do species of PE with shorter side chains, whereas PE with longer side chains decrease. PC and PI are structural bilayer forming lipids. PI is also a reservoir for secondary signaling molecules, which take part in stress signaling. Conically shaped PE imparts negative membrane curvature and is prone to phase transition at high temperatures, introducing lesions and damaging membrane integrity. Shorter acyl tails are reported to increase this transition temperature in yeast, and perhaps the remodeling seen here may be to support the bilayer structure during heat stress. 15 cardiolipin species were detected by MS1 mass and chromatography. During heat stress, the relative abundance of longer chain cardiolipin increases and shorter chain decrease in the heat-sensitive wheat variety. Longer acyl tails encourage negative membrane curvature, causing curvature stress, which can be overcome through incorporation of membrane proteins. So one possibility is that cardiolipin remodeling in the heat-sensitive wheat variety is for the purpose of incorporating respiratory complex proteins to meet the energy needs. Spectrophotometric assays measured respiratory complex activity. During heat treatment, the activity of complex 1 significantly increased in the heat-tolerant wheat variety and significantly decreased in the heat-sensitive variety. Protein incorporation into the membranes relies on the hydrophobic matching between the amino acids and the acyl tails of the lipids. In E. coli, the activity of complex 1 is reduced when there are changes in the lipids that are present during the assay, because specific lipids are needed to insert and stabilize the protein in the membrane. So complex 1 is a multimeric protein, and the changes in the lipid profile may impact on its assembly and thus the activity of the complex in the heat-sensitive variety, or alternatively, the abundance of the complex in the membrane may have changed, which may explain the changes in the activity. So in summary, heat sensitivity in wheat is evident at the seedling stage. Mitochondrial lipid remodeling varies between the wheat varieties, and this may affect the lipoprotein network with the complex 1 activity decreasing in the heat-sensitive wheat variety. Thank you.
Thanks, Aigul. Please ask your questions. There's a QA session coming up. So the next speaker, Gabriel Onsky from Baker Idea Institute from Melbourne. I think Gabriel is going to share his screen. So
yeah.
Sharing.
Can you all see my screen?
No.
Ah, sorry. Now you should be able to see it.
Perfect. Thank you.
Okay. So today I'll be talking about the detection and removal of early sources of unwanted variation. And so essentially in this work, I'm going to be looking at the unwanted variation introduced in the early stages of the experiment. So not so much concentrating on instrumental machine run and sample preparation unwanted variation, but more so in the unwanted variation that we don't generally measure and we don't generally have controls for. So here we use a targeted approach, but essentially there is this red area, and in large cohort studies where we have thousands of samples, it's often collected over several years in several collection centers using different equipment and sometimes even different protocols that are not generally considered when we normalize the data or try to remove unwanted variation. So we can remove the unwanted variation here fairly well, but not so much the earlier variation. So for the purpose of, so here I'm going to explore the Bastleton dataset, which is quite a large population cohort in which we have the plasma samples from almost 4,500 people. And the samples were collected about in 94, 95, and then extracted about 20 years later. And it's very important to note that the samples were randomized prior to extraction and machine run. And that's very important because it actually lets us identify what unwanted variation might be associated with the early sample collection and handling, as opposed to the later stages, because there's no confounding. And the data I'm going to be showing has been normalized using internal standard to get the concentration, and then batch was centered using the QC samples. So here, to assess and find the presence of this unwanted variation, you can look at PCA plots. So here, doing a PCA on the log transform data, and rather than plotting PC1 against PC2, I'm plotting the PCs against either run order or collection order. So after removing unwanted variation or strong unwanted signals, we don't expect there to be any association between the top signals and either run order and collection order. As we can see, there is no association between the top PCs and run order, but when the samples are ordered in collection order, they're clear signs, they're clear artifacts, and we can see the clear changes that are associated with different times of the collection process. And these are signals that are generally unwanted, and they're quite strong. So the top 3 PCs correspond to about 50% of the variation. And in all 3 PCs, we can see quite clear unwanted signal. And that's something that's going to affect any downstream analysis we might want to do with this data. And again, that's all comes from the red area and not so much from the green area. So of course, if we detect some early stage unwanted variation, we want to remove that variation. But we should always consider that the unwanted variation might be confounded with some biological signals. So here I'm plotting the age of the participants against samples in collection order to essentially assess whether all we're seeing in the sort of early stages unwanted variation is actually unwanted and due to different maybe collection and handling procedures, or maybe some of it is actually due to some biological differences. And here there's clear biological differences as well. So we need to be very careful when we're trying to remove this variation. But essentially the general strategy is to regress out known biological signals from the data. We then perform PCA on the residuals and select the top PCs that show clear unwanted variation. So clear structure unwanted variation usually will be 1 or 2 PCs. We then remove the signals and use the corrected data for some downstream analysis. But in any normalization, it's crucial to assess if we actually timing out ahead and if it actually helps us for downstream analysis. And here in the Bastleton data, we had some genomics information. So we have SNPs, and they do not contain any of this early unwanted variation. So if there's an improvement or increase in the association between lipidomics and SNPs, it's generally a good sign that we have removed some unwanted variation. And clearly here, we have the original heritability and the corrected heritability, and we see a great increase in the heritability. So there is an increase in the biological association of the corrected data. And when we look at the GWAS significant hits, there's also an increase in the significance following the normalization. So essentially, that removing this early unwanted variation allows us to improve the power and potentially minimize false discovery due to this early unwanted variation. So that's essentially what we're trying to do, and it's quite important to check that in large cohorts. Thank you.
Thank you, Gabriel. So share my screen now.
Yeah.
So our next lightning talk from V. Heine from Duke News NUS Singapore. Take away, Yati, you need to share your slides now.
Sure. Yeah. Can you see my screen?
Yes, we can.
Hello? Okay, sure. Okay. Hi, everyone. Very good afternoon to you. I'm Heine, and today I'm going to share with you a review of kynurenine pathway in renal diseases. So the kynurenine pathway is an important catabolic pathway for converting tryptophan to NAD+. During inflammation, the kynurenine pathway is activated, and some of its metabolites, such as kynurenic acid, are anti-inflammatory and can act as counter-regulatory mechanism to suppress inflammation. During our research, we found that the kynurenine pathway is involved in many renal diseases, especially chronic kidney disease, AKI, and kidney transplant rejection. And so for the rest of my talk, I'll be looking at these diseases. First, acute kidney injury, also known as AKI, is a short-term impairment in kidney function. The causes are numerous, but regardless of the etiologies, AKI generally follow these three phases in which cell injury evokes an inflammatory response by the body that is ultimately followed by a period of repair. And since inflammation can induce kynurenine pathway, it is not surprising to see that in many AKI patients, the kynurenine metabolites are increased, and the increase often correlate with inflammatory markers and also occur quite early on during the disease. Other studies have looked at whether manipulating the kynurenine pathway can help with AKI. And it has been shown that the inhibition of KMO, which diverts the kynurenine pathway towards the production of kynurenic acid, and the inhibition of ACMSD, which increase the production of NAD+, can help with various models of AKI, as demonstrated by improvement in renal function, as well as better histological scores. And these beneficial effects have been attributed to the metabolites that were increased. Moving on to chronic kidney disease, where uremia is often a clinical feature. Well, incidentally, the kynurenine metabolites belong to what they call the protein-bound uremic solutes. So what this means is they rely on the transport of renal transporters for excretion. And in chronic kidney disease, where the proximal tubules are often a major site of injury, the excretion of kynurenine metabolites are impaired, and they tend to accumulate in the body of patients, as seen in these observational studies. As the kynurenine metabolites increase in the body, the question is, are they harmful to the body? Well, on one hand, you have compounds such as 3-hydroxykynurenine and quinolinic acid that seems to correlate with the with the worsening of anemia and atherosclerosis. On the other hand, you have compounds such as kynurenic acid and picolinic acid that seems to have beneficial effects on complications such as hypertension and osteoporosis. So as to the question of what is the overall effect of kynurenine metabolites on complications of chronic kidney disease, the jury is still out, and this area of research is still ongoing. Lastly, I'll look at the kidney transplant rejection. Given the importance of kynurenine pathway as indicator of inflammation, it's not surprising to see that many of its metabolites can predict rejection and correlate with inflammation and mortality. And for the last part of my talk, I'm just going to share with you the importance of kynurenine pathway in the immunosuppressive mechanism of the drug belatacept. So belatacept, also known as the CTLA-4 immunoglobulin, is a T-cell co-stimulation blocker. It works by inhibiting the interaction between dendritic cells and T cells. But unlike other T-cell co-stimulation blocker, it also has a second mechanism, which is that it can increase the kynurenine pathway by inducing IDO. And the activation of kynurenine pathway is found to be essential for its therapeutic activity. The inhibition of IDO, whether pharmacologically or through gene knockout, has been found to abolish its beneficial effect, while patients receiving belatacept have been shown to have an upregulation of IDO in their peripheral blood. The fact that belatacept is currently the one and only co-stimulation blocker approved by FDA for use in kidney transplant rejection goes to show the importance of kynurenine pathway in the immunosuppression of kidney transplant patients. So in summary, we have looked at the importance of kynurenine pathway in acute and chronic kidney disease. And our lab is quite experienced with monitoring of kynurenine metabolites, especially in large cohort studies. So if you're interested to work with us, you can please feel free to contact my PI. And I'd just like to take this opportunity to thank the organizers for giving me the chance to speak, and also you, the audience, for your kind attention. Thank you so much.
Thank you. Very nice talk, Henning. So we'll move to the next one, next slide. Sorry, I'm a bit slow. So the last lightning talk today, Alessandro, Pizzit, you're from Italy. I'm sorry for you had to wake up early, so please take away and share your slides.
I'm not allowed to share, however. It says this, okay, this will stop, yes.
Yeah, you can share?
Yes, I believe you're now seeing the slides.
In presenter mode.
Yeah.
Slide sorter mode, I should say. Yeah.
Is this
Great.
okay? Yeah.
Yeah.
Perfect.
And just move my face out of the way. Give me a second here. Mind. Do it this way then. I'm just going to try and move.
That's good.
That's good.
No, even worse. Right, well, let me just jump into this. So again, thank you for the opportunity to talk to today. My name is Alessandro. I'm one of the field application scientists for Metabolon's international business team. And the following COVID-19 case study was run by a group of researchers from Dian in China using Metabolon's licensed technology. And it exemplifies how metabolomics can provide unique biological insights supporting the profiling of infectious disease patients. The study addresses the need to better understand SARS-CoV and to develop effective treatments. The work, this beautiful work, was recently published in Cell, first author Shen et al., and the title is Proteomic and Metabolomic Characterization of COVID-19 Patient Sera. And as I know that everyone knows here, the pathophysiology of SARS-CoV-2 varies dramatically amongst individuals. Some individuals are asymptomatic, others develop severe disease, resulting in a significant morbidity and mortality. Now, the aim of the study was to identify biomarkers that could predict which infected individuals would develop severe COVID-19 infection. And the authors used serum metabolomics and proteomics to differentiate COVID-19 infection from other respiratory infection in healthy controls. Changes in metabolite profiles were found to correspond to alterations in inflammation and in the immune response. What you see in the graph in the top center of the slide is the uniform manifold approximation and projection of sera samples using 847 metabolites. Serum from patients with COVID-19 revealed significant alterations in amino acid metabolism, inflammatory mediators, and glycogen synthesis metabolites. These metabolites can be associated with macrophage dysfunction, inflammation, and platelet function and clotting factors. And again, in the center top right of the slide, you can see the expression level change of two selected regulated metabolites with significant differences between non-severe and severe cases. In fact, metabolites of kynurenate, kynurenine, 8-methoxykynurenate were all enriched in COVID-19 patients. NAD+, the cofactor in many cellular redox reactions, can be in fact synthesized from tryptophan by the kynurenine pathway, as we just saw, and operates as a switch for macrophage effector responses. Also in the study, the researchers found continuous decrease of glycerophospholipids after COVID infection. And now it is already known that exogenous supplement of arachidonic acid can significantly suppress HCOV229E and MERS-CoV, so the Middle East Respiratory Syndrome replication. And this data suggests that severe COVID-19 patients might actually benefit from this therapeutic strategy. Now, moving on, proteomics identified increases in acute phase proteins, which tracked with alterations in metabolites, again, associated with macrophage function, inflammation, and the immune response. The researchers of this study trained a machine learning model for the profiling of sera. This machine learning paradigm using the data was then deployed to identify patients that would develop severe COVID-19 infections. And what you see in the upper left-hand corner is a summary of COVID-19 patients and machine learning design. The unrelated respiratory group is likely influenza, but was not disclosed by the researchers. And to conclude really quickly and summarize the added value that was brought by metabolomics, I would like to say that I think this study demonstrates how serum metabolomics can be used to differentiate COVID-19 infection from other respiratory infections and healthy controls. It can also be used to show how changes in metabolites correspond to alterations in inflammation and in the immune response. And finally, very importantly, it shows how the integration with proteomics allows for the development of a predictive model to identify which infected patients will develop severe disease. So I thank you all for your attention. In this short time, I hope I was able to convey how the Metabolon solution delivers integrated understanding of viral activity through metabolomics and how comprehensive metabolomic profiling can simultaneously inform on several biological processes relevant to infection, such as inflammatory and redox markers, but also eventually energetics, microbiome contribution, and xenobiotic impact. And these insights can lead to predictive response signatures and therapy strategies. So once again, thank you all for your attention and for the opportunity to present here today.
Thank you. Thank you very much. So I think we move to the question, live talk questions, Q&A.
Yep, 5 minutes for that, Tuzi.
Yeah. Any questions? Can you see any questions from chat?
I'm just having a look.
No? Okay.
I think some residual stuff for David at the moment.
Yeah, but
could I ask a question to Alessandro when Brad is trying to find the questions?
Go for it.
Hi, Alessandro. Really great talk. I'm just wondering that you were showing the sample number, like patient numbers 28 to 37. Is that a large number of samples to do a predictive model?
No, absolutely not, and it will have to be validated further. You're perfectly right about this, but it's the start of a process. Let's put it that way. And you're absolutely right about this. It's something that needs to be validated further with larger cohorts.
I could just ask a follow-up to Alessandro. The samples, when were they collected? So are they from the patients while they've got severe disease, or was this earlier on in the disease process? And because I'm wondering about, whether this is an underlying metabolic phenotype or whether it's something that develops with disease.
Yeah, so as you can imagine, the samples were collected at different stages. That's one of the challenges with this type of study. So there were different things that had to be taken into consideration, like time from onset to admission, time of progression to severe, time from admission to discharge, let alone, as you can imagine, there's also, for example, a limit in this study related to the age of patients, because the patients that went on to develop the severe form of the disease were older. So that needs to be taken into account better.
Mm-hmm. And I had a similar question for Wihar. If I was just wondering about the Kyrenin story that you were showing, and whether and I might have missed it because you went through a lot really quickly, but whether it was clear whether the changes in Kyrenin metabolites were a cause or effect of the disease.
Oh, okay. So because it can be both, because how should I put it? Okay, so for the kynurenine metabolites, it can be a reflection of the level of inflammation, because the enzymes that the rate limiting enzyme IPO can be induced by inflammation, but at the same time it is a suppressive mechanism for inflammation. So it can also work as a compensatory mechanism to suppress the harmful effect of the disease. So it's quite complicated, and if you factor in the fact that they are also tend to accumulate in the body because because they have to depend on the kidney proximal tubule for transport, and if that part of the kidney is damaged, it tends to accumulate in the body. So there are many factors that can lead to the accumulation of kynurenine metabolites, whether is it inflammation, or is it impairment of the kidney function, or is it a compensatory mechanism to suppress the inflammation. Yeah.
Thanks.
Thank you.
But do we have enough time to ask more questions, or?
Yep, if anybody's got another question, there's about a minute left.
I've got a question.
Yeah, please go ahead. Yeah.
I've got a question to Gabriel about is that was a fantastic talk. I'm always loath to use mathematical modeling to correct for any sort of error in collection or analysis. Do you think there's any way that we could change the protocols? Can you use your data to maybe inform the collection protocols so that we remove the problem at source rather than correcting for it mathematically?
I think that, yeah, ideally we would not have that problem. So if this exactly the same procedure was used everywhere, and let's say we would minimize the time of bench following the blood collection and have a very consistent sort of spin down and storage procedure, that would solve a lot of the issues. But now with some data sets that we have, or large cohorts that have been collected 20 years ago, there's not really much we can do if we want to use this data. So going forward, for sure, I think that will remove a lot of the issues we are facing now.
Yeah, we had a look at some of the samples from the RAIN study and just from the time in the freezer and saw a massive correlation with storage time as well. But anyway, fantastic talk. Really loved it. I'd love to see that in a publication.
Yes.
That's our time, Susie. Everybody's been really good. I haven't had to bang my cup for a while.
Thanks, Pat. Now I'll hand over to Anne to introduce our next keynote speaker. Anne, it's to you.
Hi, everyone. It's my pleasure to introduce our next keynote speaker, Professor Zhu Zhengqian. I hope I pronounced your name.
Yes, thank you.
Professor Zhu is the director of the Metabolomics Research Center, which is affiliated with the Interdisciplinary Research Center on Biology and Chemistry at Shanghai, which is part of the prestigious Chinese Academy of Sciences. And Jiang started his scientific career with a bachelor's degree in chemistry at Nanjing University before he moved to the United States for his PhD at the University of Massachusetts. And then he stayed in the US to work at the Scripps Research Institute, after which he got recruited back to China and established his group in Shanghai. And Jiang works in targeted and untargeted metabolomics, and always with a strong focus on developing automated data processing tools. And he has published these extensively in highly visible journals such as Nature. And today he will talk about LCMS-based untargeted metabolomics, from identification to global isotope tracing. Jiang, over to you.
Sure, thank you.
I'll stop sharing. So
not sure.
You can share, yes. Yeah.
Can you see my screen, and can everyone hear me very well?
Yeah, we can see. Yes, thank you.
Yeah, first of all, thanks a lot to the committee to organize this symposium, online symposium, and also thanks to IX for inviting me to join and give this keynote speech. And today I'm going to talk about related to the LCMS-based untargeted metabolomics, from the identification to global isotope tracing. Since I only have like 15 minutes each, and I will keep the introduction very short. I think most of you know metabolomics very well. As a short definition, it is a systems biology of the metabolites, or study of metabolites at the systems biology level. And the challenge in metabolomics is we have numerous numbers of metabolites, and it features a very high structure diversity. For example, in the human metabolome database, we already have over like 100,000 metabolites collected in this database. So it gives a lot of challenge to study all of them in one study. Therefore, LCMS-based untargeted metabolomics is one of the most popular technology for this purpose, since it's often performed with high resolution mass spec like QTOF or Orbitrap. In the data connection part, it's unbiased profiling with a very broad coverage. In one experiment, we have to collect both MS1 and MS2 data. The MS1 data is used for the peak detection and alignment, and the peak area is used for the quantification. As the first speaker has been talking about the quantification accuracy and errors. And another part is the MS2 spectrum are most time used for the structure elucidation and identification. Today, I have spent a lot of time on the MS2 part of the data process. I think one of the most challenges in on-target metabolomics is the metabolite structure elucidation. Information such as exact mass, retention time, MS/MS spectrum could be used for the structure identification. And MS/MS spectrum is one of the most recognized gold standards for structure identification. However, we also face the challenges availability for the standard MS/MS spectrum from the chemical standards. We take a simplest way. From the HMDB database, we have over 100,000 metabolites collected, but only like less than 5% of them could have available open-sourced MS/MS spectrum. But if you connect list or metany, the coverage will increase. But still, we have a lot of metabolites do not have available open-sourced MS/MS spectrum. That will present a big challenge for metabolite identification. Therefore, in current stage, I think most of the study in on-target metabolomics has the challenge I shown here. If your lab has or equipped with high-speed, high-resolution mass spec like SciEx triptol system in my lab, they can scan like 100 spectrum per second. So that means you can get a lot of metabolic peaks in your biologic samples, over like 10,000 to even to 50,000 peaks. But you can only identify very few of them, like 300 or something like that. If you look at the publications in top journal, I just select someone from like 2019 in Nature Medicine, Nature, from the cancers, from the cell lines, from the human plasma, from the mouse study. You will see that only like several hundreds of metabolites could be identified, but it's very few compared to the whole metabolome. Since we at least have like over like 100,000 metabolites in the whole metabolome. So that's why in the past several years, my group and also the whole community has spent a lot of efforts to increase the coverage of metabolite identification and annotation, including like expanding the spectrum library or creating new bioinformatics softwares to achieve the metabolite identifications. Today, I'm going to very briefly introduce the work we have been developed in my lab. We call it MetDNA. It's a metabolic reaction network-based metabolite annotation. Behind this work, there's a general principle. We think that in the KGG network or in the metabolome, the metabolic reaction paired metabolites tend to have very similar chemical structures and also MS/MS spectrum. Here I give an example from the aspartate, between aspartate and N-carbonyl aspartate. They share very, probably I can get to the, I'm not sure whether you can see my arrow. Those two metabolites, they share very similar chemical structures, and also they have very similar MS/MS spectrum. And we have a systematic and large-scale investigation from metany from this database. We see that with the chemical structure similarity increases, and the spectrum similarity also increases. So based on this principle, we developed a bioinformatic tool. We call it MetDNA, metabolite identification and dysregulated network analyze. The MetDNA tool has developed and employed a principle we call the surrogating approach. This surrogating approach leverages the MS/MS spectrum of known metabolites for unknown metabolites identification. I take the asparagine, this is amino acids, as an example. We do have the standard MS/MS spectrum. We also identify this metabolite from the biologic samples. And we use the MS/MS spectrum from the asparagine to achieve the identification of a total of 28 reaction paired metabolites in this network. And all those metabolites has very high similar chemical structures with asparagine. This approach, surrogating approach, overcomes the limitation of availability of standard MS/MS spectrum, which in other words, which means even you do not have this standard MS/MS spectrum available, you still can use the spectrum from this chemical structure similarly metabolite to identify them. we do not only use this surrogating approach at one time, we designed a recursive analyze algorithm in the MedDNA to reapply this surrogating approach many times. If in the round 0, we have a certain number of metabolites has been identified using the spectrum library, and we put it into a network to identify their reaction paired metabolites. Once those metabolites has been identified, we put it into the network again to identify their labels, and this goes through like one round by one round. Here you show that like we're starting from 134 metabolites and goes through like about like 21 rounds, and we can expand the coverage to like about 1,300. Then this coverage can go through by about increased by about 5 to 10 folds. Put all positive and negative data together, MedDNA enables the large-scale metabolite annotation by about 2,000 metabolites can be annotated in one experiment. And this work has been published in like 2019 in about April, and we also provide a free web server. You can visit from here, meddna.zulab.cn, to see the to use this bioinformatic tool. Over like about 1 year, and we have already have like over 800 registered users from more than 30 countries and territories, and we have helped them to finish about like 6,000 projects. After that, I was still thinking the conventional on-target metabolomics, most of the measurements are to measure the peak area or intensities. This peak area or intensity represents the concentration or either absolute or relative concentrations in the biologic samples. If we compare that to the cars on the road, you will see that this concentration represents as the how many cars on the road. But in the other side, as a comparison, we also care about the traffic, how car flows on the road. So that's compared in the metabolomics analyze, we call it the dynamic analyze or flux in the analyze. So we think that on-target metabolomics, we should go to not only measure the concentration or relative concentration in a snapshot of the biologic system, we also need to reason out why the concentration is high or why the concentration is getting lower in a certain biological system, like a gene knockout or certain like a biological phenomenon. So if you want to do the dynamic analyze, you have to introduce this step isotope tracing analyze. Actually introduce the carbon-13, most time is carbon-13 label tracer into the biologic system, and to tracing the labeled metabolites, where that goes, and to quantify the biological pathway activities. This technology has been widely used in the biological study in the metabolomics analyze, but it has a limitation. It's limited to a few like known metabolites, and with limited scales. Most cases you can analyze like tens, and most times less than 100 metabolites in that scale. But as I said, in the on-target metabolomics, we always tracing like we need to trace like up to like thousands of metabolites in one study. That's why in the very recently we developed this new tool, global isotope tracing in on-target metabolomics. We integrate the on-target LC-MS analyze for the high coverage measurement of metabolites, and we use MedDNA for the metabolic reaction network-based metabolite annotation. And in addition to those two, we developed a new bioinformatic tool we call the MedTracer for the global isotope tracing to measure metabolic activity and dynamics. This work hasn't been published. We are wrapping up, but very briefly to introduce the processing steps. We divided the MedTracer processing steps into 3 major steps. The first one is generation of isotope log target list from the MedDNA annotated metabolites. And the second step is the extraction of those generated isotope log peaks from these labeled biologic samples. The third step is to perform the natural isotope correction and to calculate the mass isotopal distribution calculation. We call it MID calculation. And once you have that, here you give an example in the cellular tracing study, we switch cells from the normal culture to carbon-13 tracer condition. We use LC-MS to measure, actually in my lab, we use Triptop 6600 system to measure all the metabolites all together. And we use MedDNA to annotate them, can annotate over 1,300 metabolites. And we use MedTracer to identify the metabolites incorporated with the carbon-13 tracer. And to quantify the labeling extent, we define as MP, moles percentage enrichment. That means how many atoms can be allocated, belongs to the carbon-sorting metabolite, carbon-sorting glucose metabolites. And this can help you to reveal nutrition dependency and pathway activity. In this cell study, actually, we found that a total of 784 metabolites could be carbon-sorting labeled. And before we jump into the biology, we also do some performance benchmark with other existing tools, including Marvin from Professor Josh Novino's lab at Princeton University, and X13CMS from Gary Susak and Gary Petty's lab, and GORGE from Professor Oscar Yan's lab. And we found that in metabolite targets level, isoblocks level, or level metabolite level in different calculation, MetaTracer has a significantly higher coverage in isotope tracing analysis. We also analyzed the reproducibility on the data extraction. We compared in both isoblocks level and metabolite level, MetaTracer has significantly higher reproducibility compared to the Marvin. That means in analytical performance, the MetaTracer has a better performance than other existing tools. After that, we put this test this tool in the in vivo tracing using Drosophila. Actually, it's a fruit fly animal model, most time used for genetics and aging study. And we feed the fruit fly with carbon-sorting labeled glucose and collect samples at different time points. And we found that in the head of the fruit fly, we could find 429 metabolites could be labeled with carbon-sorting. And we class them into 3 different groups, including a high labeling red group, medium labeling red group, and low labeling red group. And for each of the labeling metabolites, we introduce the kinetic flux profiling. Actually, at different time points, we use the exponential function to fitting the labeling curve. We found that actually they will generate a k value the k value indicating the relative flux. That means here we do not do the absolute quantification, so it's relative quantification, so it's a relative flux. It also characterizes the tracer incorporation rate from the carbon-sorting glucose. So that's so the k value indicating the tracer incorporation rate. From the tracer incorporation rate, we quantified this over 300 labeling metabolites and ranked from the k values, and by dividing them into 3 groups, including high labeling rate, the k value is around 0.7 to 8, and the medium labeling rate, the k value is around 0.2 to 3, and low labeling rate is about 0.1. And we map those metabolites into different pathways. It's about 17 different metabolic pathways. You will notice that the glucose metabolism, amino acids related and fatty acid related, they're through from high labeling to low labeling. Here are the examples for the different labeling metabolites. It's very high labeling rate and low labeling rate. I think that's the last slides from my talk. Is a summary. We think that LC-MS-based on-target metabolomics enables to measure metabolites in a very large scale, and identification is a challenge. Here we developed bioinformatics tool, MetDNA, to identify about 2,000 metabolites in one experiment. Stable isotope tracing enables to measure activity and dynamics is an important part of the metabolomic studies. MetaTracer provides a global isotope tracing in on-target metabolomics for measure the metabolic activity and dynamics. Finally, I would like to thank my group and the funding, also the support from the SciEx. Actually, we do have like 2 instruments from SciEx in my lab. Thank you all.
Thank you very much, Professor. Very nice talk. And questions for?
There is a question in the Q&A box, Tussi.
Okay.
I see the MetDNA software used to identify human metabolites only. No, we integrate the KGG database into the MetDNA software. So any metabolites or species included in the KGG database, they can be identified and performed with the MetDNA software. So on the software web server, you can choose the species from your sample. You can choose that. Yeah.
Is there anyone waiting? I'm happy to ask one question from you, Professor. Are you planning to expand that to lipidomics area, the labeling and incorporation?
Yes, I think that's a very good idea. We do, in current stage, we do use Triptop 6600 for the general on-target lipidomic studies. And yes, it's an easy step forward to treat that as like isotope tracing study. But lipids, they may have some very special treatment because the M+2 peak always have overlap from the different pressures. So we need a special algorithm to treat that. Yes.
Yeah. Thank you.
I have a question. So
go for it, Peter.
Thanks for the talk. That was excellent. Actually, I have two questions. The first question is about validation of the metDNA approach. And so you identify a lot of metabolites, but have you been able to validate those by an independent pathway to the identification? And the second part of the question is really, if you integrate the MET tracer with the metDNA, do the different pathways that you can identify with the MET tracer agree, that is high or low incorporation rate, agree with the annotation that you give to the metabolites, because you'll expect, metabolites on the same pathway to show the same level of incorporation?
Yes, I think Peter have a very good question. The first question is the validation of the annotation in metDNA. In our publication, we do a very different approach. We employ different approaches for the validation. The most one is just to use the external database, like from NIST, from METNI, to generate those annotations and compare with metDNA. And later we also compare with some other bioinformatics tools like Series or CFMID to see that. From that part, actually, that you can see more details in our published papers. And in the same time, yes, at that time, we're also thinking whether we can introduce the carbon sorting labeled the data set as a validation for the annotation. That's why we moved to MetaTracer work. And slowly we found that in the MetaTracer work, we can see a lot of metabolites can be correctly labeled. And in that same pathway, these results as also can be partly used to validate whether the annotations from metDNA are correct or not. That's also the very good point, and we're also doing that too. Yeah.
Tusi, we do have one more question.
Yeah.
I see the question. Carbon sorting labeled glucose is quite expensive, so your rat or other animal model have to eat carbon sorting glucose all the time. Yes, it is very expensive, but compared to other tracer like carbon sorting labeled glutamine or something else, it's relative affordable. And yes, in the Drosophila study, we do let the Drosophila to eat the carbon sorting labeled for up to like 24 hours. But Drosophila is very small, so that's why we do not use the mouse or rat for the study as a biology or technology development. It's very costly. So Drosophila is very small. The cost is not very high. Yes.
They don't eat much, huh?
They don't eat much.
Very good. It reminds me of the old fly in my soup joke, but not enough time for that. Over to you, Tusi.
Yeah, thank you very much. So I'll share my screen again for the next talk. I'll hand over to Fana to introduce Professor Yu. It's my pleasure. Fana, take
Thank you, Tusi. Thank you. Again, it's my great pleasure to introduce Professor Yu Jia. She's from Department of Chemistry, Tsinghua University. I apologize if my pronunciation is wrong. Professor Jia actually did her PhD from Purdue University. After she completed her postdoc there, she actually worked there in the Department of Chemistry from 2009 to 2017 before coming back to China. Now she's actually using a radical chemistry as a unique approach to achieve enhanced bioanalysis using mass spectrometry. So today, Professor Jha is going to talk about new capabilities in resolving isomers for lipid analysis. So Professor Jha, it's your floor now.
Thank you, Farhana, for your nice introduction. So I'm going to share my screen. Okay. Okay. Can you see my screen?
Yes.
Is it in like this full screen mode or? Yes, okay.
Now it is. Now it is.
Good, great. I'll get my laser pointer here out. Excellent. So I really appreciate the committee organizing this nice series of virtual podium. So I have not traveled outside Beijing since February. So this is a great way of seeing other and exchange our science worldwide. So today I'm going to update some of our recent research involving developing techniques for seeing detailed structure of lipids. We all know that lipids have a lot of great functions and diverse functions, and those functions are supported by their diverse structures. Using glycerophospholipids as an example, due to the various head groups and the combination of different fatty acyl chains and how they are organized or sorted onto this SN1, SN2 position of glycerol backbone, as well as many different fatty acyl chain modifications, including desaturation, oxidation, the methyl branching. So actually, there's a lot of different variety of molecules over there. However, we don't have enough standard. And it's impossible for us to synthesize all the standards. So there's a strong need to develop a method that can independent ID the lipids in the biological system. So currently, the available tandem mass spectrometry techniques is very good in providing the class information, which tells the head group, as well as the fatty acyl or ether chain composition. But it cannot tell you more about the detailed structure information, especially on the fatty acyl chain modification. But we know they are quite different molecules. They should have different functions. So it's important to know them and identify them. So there's a surging kind of research interest in the mass spec community, as well as in the lipid analysis community in those detailed molecular structure. I would suggest you guys to read a recent perspective by Shane Ellis group and co-workers. They suggest it's very important to go to the details of lipid structure. And in that perspective, they demonstrate the emerging new tandem mass spec techniques, which are capable in identify the location of double bond, as well as in differentiating or even quantifying the coexisting double bond isomers in the mixture without using any standards. So those are UVPD. This is developed by Braubel group and the Rate group, and the Pertuna-Bucci modification coupled with tandem mass spectrometry by my group, as well as, can I turn this off? It's blocking my view. Oops. Give me 1 second. How can I turn the video kind of screen
If it
off?
helps, we can't see it.
Okay, it's blocking a little bit of my screen. Anyway, I'll go with that. So, and then this, I could not see Blankspeed's photo here, but anyway, so also ID developed by Blankspeed's group. Those all three techniques are still developing, so I don't want to compare which one is the best, which one is good or bad. I just want to say that because Pertuna-Bucci reaction is a solution modification or modification outside mass spectrometer, so it's more available to commercial instruments. You don't need to buy a specialized instrument to do the experiments to see CC double bond. So how does it work? So Pertuni-Bucci reaction is a photochemical reaction. It involves the excited state of a ketone or aldehyde. So once excited, this ketone aldehyde will do cyclic addition onto a cis double bond. So this has a high selectivity. So when we first tried the reaction, we use acetone as a PP reagent. It can also be used as the ESI solvent. So we just dissolve the lipid in this water acetone solvent, use a mercury lamp to irradiate this solution, and within 10 seconds, you'll see the cyclic addition product. Now we have changed the cis double bond functional group into 4 membrane. So this creates a much better chance we're using CID to cleave this ring and forming this kind of a cis double bond diagnostic lines, because they carry the original information of the cis double bond. So this can be applied to monounsaturated fatty acids as well as for polyunsaturated fatty acids. And just want to add one note for polyunsaturated fatty acids. Even we have several cis double bond, but the PB reaction generally does not have selectivity toward one specific double bond. So you'll see we generally have quite even diagnostic intensities from one saturated fatty acid. So we can also use the double bond diagnostic lines to do relative quantitation and also absolute quantitation, as shown here. So since the first experiments, 2014, my group is focusing on expanding the scope of PB reaction from free fatty acid to conjugated fatty acid to those kind of to phospholipids and to neutral lipids as well. And we have also been active searching for better PB reagents other than acetone. I'll show you why. And also, our ultimate goal is to make sure this PB tandem mass spectrometry strategy can be implemented in all existing lipidomics workflows. So people all around can know what kind of unsaturated lipid they are working with. And I also want to share with you that we are not alone along the quest. There are several groups, important groups that have been joining us and making important developments. Here, I want to highlight Professor Hyles' research. He pioneered the incorporation of PB reaction with multi-imaging. So you get fantastic high resolution of the tissue with double bond specificity. So in 2019, we have streamlined the acetone PB reaction onto the LC system. The key point to that technique is that we use acetone as a mobile phase, and we use a highly efficient flow reactor connected between the LC and the ESI source of the instrument. And then we can do tandem mass spectrometry, and we also have developed a software, not powerful as Zhenjiang's, but it can annotate, like do independent annotation of the lipid structure to the cis double bond location level, as well as to do relative isomer quantitation. So we have also worked with a company called Perspect, and they have commercialized this reactor called Omega reactor. This workflow actually gives broad coverage of phospholipids. Typically, we get like 90 to 80% coverage of what you usually see from LC-MS from fatty acid level. So that will give above 200 lipids molecular structure identification down to double bond locations and many groups of isomers. And recently, my student Xiatian, and he found that if you use methylation, phosphomethylation can further improve the identification for PG and PI. I believe this workflow is readily transferable to different labs. So my undergrad researcher, Henning, at that time is junior, was hosted by Sling. So he brought the reactor, a helicolumn, and analyzed the lipid extracts from 4 types of different tissue and plasma from mice. On the typical level of those different types of biological samples, he get like about 200 identification of phospholipids. And for this large amount of sample, you can do Here just show a snapshot of what can you get. If you're interested in a specific molecular species like PC 160, 161, you can do a cross comparison in different types of tissue and samples. And here you can see that for brain tissue, where FAST2 expression is relatively high, and indeed the N10 isomer shows relative high concentration. However, this is a different situation for 18-1. For 18-1, even for the same tissue, we don't see its corresponding N10 isomer, which suggests the synthetic pathway for 18-1 is quite different for choosing different isomers. So acetone is a great PP reagent because it's highly compatible and it's clean to mass spec. However, we did realize it has several shortcomings and would like to look for newer generation of PP reagents. For example, it will have higher absorptivity, like acetophenol will have 10,000 times higher absorptivity than acetone, and longer excited lifetime, which means higher yield and limited side reaction, more stable side reaction, and especially larger molecular weight, which can separate the reaction products profile from the unreacted lipids so as to reduce the chemical interference. So the best we have found is called TriFab, so the trifluoro substitute acetophenol, and it offers a limit of identification for this standard compound 10 times lower than acetone, and also reasonable yield. An important lesson I want to share with you is that although you might have a very nice peak from the reaction, you think it's a PB product, and actually it contains two types of major side reactions due to cross-linking. This is all radical chemistry. So the PB products actually here can vary from 20% to 70% of the total peak intensity. So that's an important lesson we have learned. You cannot just assume what you see over there, the mass spec peak is all the PB products. So for the TriFab, it offers obvious advantage for shotgun analysis. We can see that it gets this comparison to acetone, it gets reasonable reaction yield, and most importantly, we have this two population of the PB products versus unreacted lipid, so makes our later tandem mass spectrometry data much cleaner for identification. And then this advantage, TriFab is also quite highly compatible with different types of CC double bond. Here shows conjugated, the value ether CC double bond. And when we do offline coupling of the derivatization with LCMS, we got similar identification or performance in terms of the lipid double bond coverage and number of identification. So this is also good message because that means you don't need to mess up around with your LC separation system. You can directly use what's reacted in the vial and injected in your already optimized LC for lipid identification. So analysis of neutral lipids is another challenge because we need to worry about ionization efficiency. And Heine's group also have shown that if you use 3-acetylpyridine, it has a capability of improved ionization for fatty acids and neutral lipids. And we set out the quest of testing different position substitute acetylpyridine. And we found that actually the two substitute acetylpyridine has a higher reaction yield than the 3-acetylpyridine, and also is more pure for the PB product. So it can improve the detection, also improve the detection, even though we don't have 100% yield as compared to intact CE by some 8 times, I cannot see here, I guess, and also improve the detection of the double bond, like 100 times the acetone PP reaction. Also, we have a great linear dynamic range for quantitation of isomers. So Xiaobo, she developed this offline 2-acetylpyridine reaction coupled with reverse LC tandem mass spectrometry. And this is a record high number of identifying 27 unsaturated CE, which spans 10 orders of magnitude in concentration. And most important, you can see here for the 6 groups of double bond isomers we have identified, many of them contain multiple double bond isomers, and of low concentration, like relative concentration of 5% or less. 2-acetylpurine is a magical reagent. So my collaborators, Professor Xiaoxiao Ma and Ouyang, they found that if you use 2-acetylpurine for phospholipids, and you choose to do a PB reaction and the MS3-CID in a single spectrum, you can see the double bond location as well as the SN position of the acyl chain. So that's fantastic. So now we know we can do double bond isomer analysis and SN isomer analysis. So what can we do with the information? So in fact, the isomer composition reflects the relative enzymatic activities in these metabolic pathways. So if there's a phenotype or disease condition which dysregulate their corresponding pathways, the change of isomers will very sensitively reflect that. So as recently shown by Varens and co-workers, for FAST2, for the certain cancer FAST2 is increased, you'll definitely see an increase of its corresponding desaturated monounsaturated fatty acids. Okay, 2 more slides maybe. So in our recent 3 papers, we have shown that lipid isomer ratio can be used as a new types of biomarker. There's a great advantage of measuring the isomer ratio, as we have learned that if you measure the peak intensity or peak every year, there's always quality control issues, right? But if I measure the relative ratios, the peak ratios, we found there's very small interpersonal variations. It's typically less than 15%, and you don't need to add any internal standard. It's great for biomedical research. And sometimes if you just measure double bond isomers, it's good enough for phenotyping. Sometimes SN isomer is good for phenotyping. And in this case, I want to highlight that you need to have both information and you get much better phenotyping for the human lung cancer. So for this short talk, I just want to convey 2 pieces of message. First, I think the PB tandem mass spectrometry technique is ready to be transferred to other lipidomic research labs and to enable people to see more detailed structure information of lipids. Secondly, I think the capability of measuring the isomer ratios, no matter it's double bond isomer ratios or SN position isomers, it provides a unique capability for disease phenotyping. Okay, and finally, I would like to thank my group. This is taken just after 8 months kind of like lockdown of Tsinghua University. Students were just back on campus with the Hemingway and catching a fish, actually a fish skeleton here. It's been blocked by us. And I would also like to thank my collaborators. And here, there's many ongoing projects about lipid research. We're still collecting data. And hopefully I can share those with you in future interactions. Thank you.
Thank you very much, Professor. We got one question for you. Priyanka Reddy, she's asking, what is the % of lipids that can be leveled by acetone?
Oh, 30% at most. Not very high.
Is there any other questions?
How can I quit the screen sharing?
We can take it back off you.
Okay.
Tussie, can you reclaim the screen?
You have another question from Corey. He's asking, is the PB reaction with tri-FAP consistent across lipids regardless of fatty acid constituents?
You mean the reaction yield consistent across lipids? I did not catch the early part of the question. I can now read.
Yeah, you can, yeah.
Okay. From T. Kim Boom, is that number range to consider small, medium? No.
Corey.
Corey is saying that yes.
Okay. With tri-FAP consistent across lipids. The consistent, no, it has different reaction efficiency for the electron-rich double bonds. For example, vinyl ether, its reaction efficiency will definitely exceed 30%, it reaches 50%. So it varies for different types of CC double bond. But for isolated CC double bond, typically we observe 20 to 30% of reaction yield.
One more question. I see that charged PB reagent like acetylpyridine and tri-FAP may have higher yield. Which one you think is better?
Oh, so I probably didn't explain plain and clear. For acetylpyridine, I think it's best for neutral lipids because you can see the double bond location at the same time improve the ionization. And for tri-FAP, we use it quite often for phospholipids, which are already well charged. So it has different kind of applications.
Yes.
Really nice talk and very interesting potential applications, I think, for this technology. And I'm wondering about the clinical type applications. And so have you looked to see what the throughput is for, or what throughput you can get for this type of technology in terms of samples and how large a cohort you could run?
Yes,
and still maintain, I should say, still maintain that sort of precision, at least in the ratios.
Oh yes, currently we because it's a solution reaction. So let me use an offline reaction as an example. So you use a certain amount of lipid solution extract and do the reaction and inject into the LC. We add also in terms of adding the time, we do add one more run than the traditional kind of LCMS because typically we need to know the class information, the fatty acid information, and then we add this PB reaction to nail down like on which fatty acid we have this CC double bond location. So other than that, we did not add any other additional time in terms to slow down the throughput.
Okay.
So that's time for questions following the keynote. Tussie, I'll hand it back to you momentarily.
Yeah, thanks, Brad. So we're coming to end of our fourth session. So I'm pleased to introduce Professor Peter Mikkel. Peter is instrumental to lipidomics to my work. Peter is the head of domain of the IDI for Metabolomics Laboratory, and he's the editor-in-chief for Metabolite Journal for the International Metabolomics Society for the metabolomics. If you are, I think all of us know Peter. So it's a privilege to introduce you to give the closing remarks.
Okay, thanks, Tussie. Yes, I was going to say, could you take that photo down and could you put up the photo of the organising committee?
Okay.
No, not that one.
Yeah. I need to go back, but not sure.
What I would like to do first is to acknowledge the organising committee for the great effort and the great work they've done in bringing together this series of virtual podiums. No, back a couple more, I think it is, Susie. That's the one. That's the one. They're a little bit stretched in some of those, Brad. I see. Put on a bit of weight.
I'll have to fix that one up next virtual podium.
That's what lockdown does to you. But look, it's really been all of these people contributing together that have been able to bring this together, but particularly Tussie, who was really the instigator and leader of this. And so I think if we were all sitting around an auditorium, this would be the time where everyone would clap and thank Tussie and the other organisers for their effort. And I think it's also, this has been a great opportunity for us to come together in what has been, a very difficult time all around the world. And so over this last 4 weeks, we've really heard from some of the leaders in the field of metabolomics and lipidomics. And at the same time, we've been able to hear from some of the newest members of the field of metabolomics and lipidomics, and I think that's been a great mixture. And these talks, as we've heard today and in earlier weeks, have really covered the technology, the analytics and the biology of metabolism in humans and well, everywhere from humans down to plants and microbes. And so I think has demonstrated the breadth of the field, but also how active we are in the Asia-Pacific region. And so while in these times it's difficult to time things so that we can link easily with the US or with Europe, I think we've demonstrated that we have a very vibrant and a very active community here in this Asia-Pacific region. And, you know, and we've had great talks from Singapore, from Japan, from China, from Australia. And I won't list them all, but you know, we were all listening to the talks over the past 4 weeks. So the final thing that I would like to do is thank all the participants who've logged in each week and listened to the talks and asked questions, and hopefully have started to interact with each other and as well as with the speakers, because I think the success of this virtual podium series is measured by firstly the benefit that people have gained from the talks themselves, but also by the benefit that they will gain by continuing the interactions beyond these podium sessions. And so I would just encourage everyone to reach out to their colleagues in their own countries or in different countries and continue that interaction over the coming weeks and months and years. So thank you all for attending. Thank you, Tusi, again for the organisation, and we'll look forward to perhaps another series of this under better conditions. So thanks, everyone.
Thanks, Peter.
Thank you, Peter.
Thank you, Peter.
Thank you.
Thank you.
Well said.
Thank you. Bye, everyone. Thank you so much.
Bye, everyone.
Have a great weekend, everyone.